Hello, I'm Jan Leifman.
I'm Gayatri Pramal Menon.
Welcome to Med News Week Conference, where we feature presentations by medicine's global leaders. Today, we have an amazing keynote speaker in Dr. Jeff Karp. Dr. Jeff Karp is the Distinguished Chair in Clinical Anesthesiology at the Brigham and Women's Hospital.
In today's presentation, Dr. Karp discusses medical innovation.
Did you know that Dr. Karp is the co-founder of multiple internationally recognized companies? He's also the recipient of over 125 patents and 65 grants, with sponsorship by multiple companies, government agencies, and foundations, including the Bill and Melinda Gates Foundation and the NFL. He has also been recognized as one of 11 Boston doctors making medical breakthroughs by Boston Magazine. He's also recognized by MIT's Technology Review Magazine, or TR35, as one of the top innovators in the world. 3 members from his laboratory have subsequently received this award after him.
He truly is a pioneer in the field of medical innovation.
So let's tune in. Let's tune in and learn from this global leader.
Thank you so much for the invitation to speak here and for that nice introduction. So before I get going, I have an institutional responsibility to declare my conflicts of interest, for which I have many. I have co-founded several companies that have spun out of my lab, and I'm an active consultant in all of them and hold equity. And today I thought maybe I would share with you some of the stories. So before I get going, I wanted to first just start off with showing deep appreciation for current and previous lab members. I feel very blessed and grateful to have such wonderful people to work with. We've had people from over 30 countries in the lab, and it's highly multidisciplinary, covering fields of engineering, all kinds of different engineering, biology, immunology. We've had a cardiac surgeon, a gastrointestinal surgeon, a dentist, and it's just highly dynamic and always changing. I really like to try to focus on creating a lateral environment where everyone can learn from each other, and really an environment where actually try to minimize the overlap in expertise, so that everybody can bring something unique to the table. And I think this really just creates a great environment. And I just want to say thank you to all the people in this picture, as well as the previous lab members. And so I thought maybe what I would do is start from kind of reverse back in time to when I was a PhD student in Toronto. And I was just really getting fired up about research. I had done some research when I was at McGill in my undergrad for a few summers. And that kind of got me hooked. And then I quickly realized during my PhD that wasn't going to be enough and that I needed to keep going. I had been following the work of Bob Langer at MIT for many years, and just really excited about all the things coming out of his lab, as well as what previous people in his lab were and current people were saying about him as a mentor. So I was, I thought, okay, I'm just going to go for the gusto here. I'm going to send him an email. Who knows whether he'll read it, but I'll just assume that he will. And so I took like several weeks to compose this email and this was in 2003 about, the subject line was a postdoc candidate with big ideas, with big in caps. And I kind of described to him how there were a couple ideas I had, and, how I would potentially go about, solving certain medical problems. And it was kind of half baked. I just did the best I could with those few weeks. And so he responded immediately, as he's known to do. And he said, basically that, he thought that my experience was interesting, but he didn't have any funding to accept me in the lab. So I thought about that, and I thought, well, maybe he's just saying that, and he just kind of wants me to go away. But maybe I should just, be optimistic and think like, okay, well, maybe if I could get my own funding, maybe he would take me. So I wrote back to him right away, and I said, well, if you give me, if I come with my own funding, would you accept me in the lab? And he immediately responded with yes. And then at the time, there was this, I'm Canadian, and there's the National Science Engineering Research Council provides fellowships to postdocs. And you don't actually have to come back to Canada at the end of it. It was 2 years of funding. So I said to him, so immediately after he said that he would accept me if I got funding, I said, would you put that in writing? And so he immediately responded, sure. So he put it in writing, and I applied for this fellowship. This was back in the days when there were no, it wasn't digital. It was like actually you print everything out. So there was no space for such a letter of support, but I just tacked it on to the end saying, if you give this fellowship to Jeff, then he'll get a position in Bob's lab. And I waited a few painful months, and then finally I got the fellowship, and I wrote back to Bob, and that's how I got into his lab. And this is a picture of us with a technology that we had developed, an adhesive technology through mimicking how geckos can walk up walls and hang from a single toe. And we were attempting to create a surgical tape that could do the same, that was elastic and biodegradable. So when I was in Bob's lab, I learned a ton of things. He was just an incredible mentor, and there were just people from all over the world working on everything you possibly can imagine. As I was advancing through my research career, I quickly realized that, I was really excited to publish work, but what was really driving me was how to bring science or scientific breakthroughs to patients, and really how could one accelerate that process. And so that's where I sort of cultivated this desire and passion for accelerated medical problem solving. And I think, we all need examples around us, in our environments of what's possible. We all need to have role models, and Bob was an incredible role model. And I had some really incredible mentors as well for my PhD, and even before then. And I think that this really helped me to kind of create a North Star of what I really wanted to do. And that was sort of seeing Bob and other people in the lab be able to take science and turn it into, products that could help patients. And so I kind of realized along the way, and this is something that, I had a lot of challenges growing up in school with various learning difficulties. I was identified as having a learning disability actually early on, and ADHD, and I kind of had to develop a lot of processes to cope with the struggles that I was kind of encountering in school. And so when I sort of realized this is what I want to do and commit to, this process of sort of creating medical products that could then, using science to then, help patients essentially, I realized that I didn't have a process for doing this, that I had been in Bob's lab and I had seen a lot of people do it. But when I started to kind of try to do it, I realized I didn't really know what to do. So it's kind of one thing to see other people do it, and it's another to do it yourself. So I realized I needed a process. And so what I did was I said, okay, well, how am I going to develop this process? I have no business training, and in fact, no formal training whatsoever in kind of commercialization of science. And so what I did was I sort of made this strategic decision that early on, my faculty position was very busy writing grants and which was actually quite difficult. I wrote over 100 grants in my first 2.5 years. Most of them were rejected. But I decided to commit to meeting people in the ecosystem, the entrepreneurial ecosystem. And I think, this is really, and I forged like a lot of, not just kind of having a network, but actually relationships, because I think in academia, because we're not trained really in how to impact the world, we need a process to do that. And so what I did was every 2 or 3 weeks for probably about 10 years, I would meet with people in the entrepreneurial ecosystem and just try to develop relationships. So the goal was, just to identify synergies with people. And this really helped me to crystallize my goal of not just publishing papers, but trying to find a way to bring those approaches forward. So just to give you a sense of what that really looked like, I would meet with patent lawyers, investors, reimbursement experts, CEOs, manufacturing experts, regulatory experts, biotech, medtech, consumer health experts, just people in the community, going for lunch with them or coffee, or just, inviting them to the lab for a tour. I would go to all kinds of different networking events and just really try to introduce myself, even though many times I didn't feel like going, I didn't feel like speaking, and, but I felt I just like, knowing that, I had kind of found this intention, this sort of passion, this purpose. And I tried to really let that lead. And so I would go to these events and be like, okay, I just have to meet like 2 new people. And I would just gradually, I was able to connect with people. And so what this did is this actually created a filter for almost like a translational filter for the projects in the lab, because now as we advance projects, I reach out to people in the community and I ask, do you think this could be manufactured? What do you think the IP space is here? Is this something investors are looking at? Is this something that, pharma or medtech would be interested in? What's the best first application, which actually is a really hard, when you develop a platform, like a material that could be used in all kinds of different things, it's hard to figure out what's the best first application to apply it. So kind of through these relationships, I was able to kind of hone in on how to make decisions about moving the science forward to maximize potential that this, if things went well, that we would be able to to bring this to patients. And so what I wanted to do is also, I spent a lot of time in self-reflection thinking about the medical problem solving process. And I just wanted to share with you, I tried to condense this down into a single slide of how we go about solving problems, and then trying to create, translational opportunities to bring those into startup companies. So I've co-founded many companies, as you saw before, and this is, I think, kind of not all of it, but a lot of the core sort of element or process that I've been using, and it's constantly evolving and being iterated. So the first step is to really consider that the problem goes beyond the medical problem. Usually, we might think of a biology problem we're trying to tackle or a medical problem, but there's the reimbursement problem, there's the clinical trial problem. For example, what's the comparator going to be in the clinical trial? And can we get that early in our experiments so that we can then determine whether our results are important or not? So often, you might see scientific data presented where you have, a graph and it looks like, what's being presented is really great compared to some of the controls. But, what we like to do is not just sort of put positive or negative controls to know whether something's working or to kind of get at the mechanism, but also controls that will tell us how close are we to the gold standard? Are we below, similar, or above? what's currently being used in the clinic. So these are some of the things. So I almost think of the problem as like a Venn diagram, and you have like the biology problem, the medical problem, but then you have all these other things like patent problem, the manufacturing problem, the regulatory problem. And so I think in order to develop technologies that can then make it to patients, we need to think about the overlap between all of these different areas. The other thing that I found is really important is thinking about what's the success threshold, or in other words, what's the best result others have achieved in a particular model, and then how much better do we need to do, and then making that the North Star of the project. So for example, let's say if we're working on like an oncology project, we might think, okay, what is the animal model that others have used, and what's the best survival they've seen for a certain time period? Maybe it's 3 out of 10 mice survived for 6 months, and so we might say, okay, well, do we need to get like double that? Do we need to double the timeline? Like, we start to think about it, and there's no sort of clear answer, but we try to make it some sort of best guess of where we think we need to get to. And then that becomes the goal of the project. And then the experiments that we conduct, we're constantly trying to conduct experiments where we're trying to learn something new and gain critical insights. And ideally, ideally we want to run experiments where we're doing these comparisons to the gold standards that are used in the clinic, if possible. It's not always possible. The other is we try to conduct experiments where we try to learn something new or something that other people have overlooked, that then provide a direct vertical for us to develop a new technology. So I just find it so easy to come up with ideas of solutions, but often those solutions are not well founded within a key insight, a key biological insight, for example. And so we look for these insights, and then we use the insights to figure out, okay, what's the solution going to look like? So I find that to be really important. And then we also try to think about our technology in terms of like, what's the unfair advantage that we have? Like, what is it that we can do that our technology can do that nothing else can do? Because I find a lot of the times we get really excited about our technology, but then we realize that there's a dozen others that can do the exact same thing. So we try then to use that as a guide to develop technologies that can do things that other technologies can't, technologies that could be enabling to get a drug to a location that no other technology can do. So that's something that we're constantly thinking about and pushing for. And then the last is what I like to refer to as translational momentum, which is, things always stall. We always run into problems. There's always challenges and failures. And so, constantly thinking about how can we maintain momentum? How can we keep being persistent? And it's not always easy, but I find one way to do it is to think about infusing energy into the project. And I think one way that usually works is finding other people who have expertise, like potential collaborators that we don't have, and then bringing them in so they can kind of look at it through a novel lens that we haven't looked at it. And this can then generate a lot of excitement, enthusiasm, and new energy. And this is not like this is almost like a continuum. It's not necessarily in this order. We're just thinking of these things constantly. And then I think the other thing too to consider is that, just as we've been advancing, that there's opportunities, sometimes we come up with ideas. And so these are kind of like, and then we look for people to collaborate with, kind of more like outgoing opportunities. And sometimes people bring us ideas and projects, and there's these incoming opportunities. And we're always trying to, create as many opportunities as we can and then apply them through these filters. But also sort of recognizing sometimes you have opportunities in front of you, just a conversation, let's say, that we just had, and that could lead to something really exciting. And I think, one of the things we try to do is really is just follow the energy. And so, there's a certain energy of collaborating with other people. And I think kind of tuning into that energy transfer between people is where it's really at. And that, that's critical, I think, to just being persistent and really pushing through the challenging times. And so let me just tell you a quick story of an example of something that happened where, I like to think of this tool of being actively opportunistic. So there was this grant. It was early in my faculty position in 2009. So I was just at it for a couple of years. And I hadn't actually been successful at all in writing grants. I had like a year left of my startup package, and I wasn't able to secure much funding. And so I applied for this grant. I think it was $40,000 to the Massachusetts Tech Transfer Center. And they had me kind of go present my grant. And it was right after my daughter was born. And like literally a few days after. So I was exhausted and, I stopped in the middle of my presentation. It just didn't seem to go well. But towards the end, as I was kind of leaving the room, somebody kind of pulled me aside and said, "Hey, let's go for coffee," one of the grant reviewers. And so I kind of thought about it and I was like, I'm busy writing grants. I really need to bring in the funding. I need to be focused. But for some reason, there was this kind of like energy that I felt, this gravity. And I thought, okay, I should go meet this guy. His name was Val Levada. So we went and met for coffee. And then he said, "Well, I think you should meet with my friend, Kathy Call at Sanofi." And so again, I'm thinking, okay, I've gone for coffee. And had a great conversation, but now it's turning into another meeting. And is this just going to be meeting after meeting? But still, I felt that gravity. I was like, okay, I'll go meet with Kathy. So I met with her, and she said, okay, well, you got to meet with Sridhar. He's not in town right now, so wait till he comes back, and he's leading this kind of regenerative medicine effort. And so I was like, okay, well, I'm getting in deep here. I need to keep going. So then I met with Sridhar and we totally hit it off. He started inviting me to give presentations at Sanofi in Boston, in San Diego, in Paris, and I started to develop relationships with other people in the company. And then he said, "Well, why don't you propose a couple ideas to us? Maybe we could fund your lab." And so I proposed two ideas. One they liked, one they didn't. And that led to 4 years of pretty significant funding. And then through the funding that I got through Sanofi, I got to meet other people in the company. And one person I met is down here in the bottom left, Bernard Gilly, who runs an entrepreneurial ecosystem in Paris. And I had shown him some of the work we had done with tissue adhesives. And he said, "Hey, how would you like to start a company on this?" And then he brought in Christophe Bancel, who's an entrepreneur that he had worked with before. This is Stéphane Bancel, who's the CEO of Moderna. This is his brother, and Christophe. And so we started a company together called Tissium, which I'll tell you a little bit more about in a moment. And then one of the people who was leading this collaboration with my lab, Jacques Delors, when he said, "Okay, I'm going to be leaving the company, let's work together." And so we actually took a technology we had developed in my lab, and we created a company called Skintifique, which is a skincare company. So this is just an example of how like a single kind of, cup of coffee with somebody led to, funding in my lab, as well as, cultivation of many really great relationships and a couple companies, which are still going strong.